There we go. All right. So the first half of the course, we've concentrated on the structure of the microbes, right? So for example, we've looked, does it have a capsule, right? And you guys are going to do this again. Probably next week we'll get a chance to actually look at those slides that you guys have been making and make these determinations for your unknowns. The second half, what we're concentrating more on now is function. What can they do, right, under certain circumstances? So a lot of the tests that we run are a fermentation, right? And so some of the ones that we're going to talk about today deal with glucose fermentation. We're going to talk about fermenting other sugars as well. So almost like the anatomy, right, and physiology of microbes as opposed to multicellular organisms. So today we're going to talk about motility, kind of review a little bit from our motility test before that we looked at last week. We just ran the methyl red and BP test. We're going to talk about the significance of those. The phenol red broth you already inoculated, we're going to look at those results, compare them to known results that I have on the table for you guys. Same thing with citrate, and then we've already just done catalase this morning, and we're going to talk about the specifics of those results and what they all mean. So, right, so as he said, right, we're going to compare this to the information that we already got on motility. The red exercises are ones that are biochemical tests, what the organisms can do, right? All these ones that we're looking at today. So when we talk about motility, the most common form of motility is flagella. And if we had the ability to stain them, we could see how many flagella they have and where they're arranged on that cell. So there's some terminology that you guys need to be aware of for flagellar arrangement. Now, of course, for your individual organism, because we're not going to stain them and look at them under the microscope, you wouldn't be able to tell what it has, what its arrangement and stuff is, unless you were to go look it up once you figure out what your unknown is, right? Which you could do. I could maybe make that bonus or something. So polar or monotrichous, trichous referring to hair-like, mono referring to one. And so that's why polar too, when we look at the shapes of these later, they tend to be rod-shaped or spiralium in shape. And so it makes sense polar at one end, right, is another term that's sometimes used for having just a single flagella at one end. Amphi refers to opposite, right, trichous. So in this case, they have one flagella at each end, right? So they have two flagella total, one at one end, one at the other end. Lophotrichous refers to a clustering, right? So where you have multiple flagella at one end or both ends. So yep, this refers to clustering. Yeah, those of you guys in my lecture, this should be reviewed, right? We went over this a long time ago. Peritrichous is completely covered, uniformly distributed flagella, right, over the entire surface. So I call it my scary cousin it. So this one we would refer to as peritrichous, right? This guy, mono or polar, right? And then notice the shape. What shape is this? Rod, bacillus shaped, right? Where these guys are spiralium. You see that? And so see how it has multiple flagella at both ends? But if it had multiple flagella at just one end, it would still be lopho. So this has one at either end, and this would be amphi, right? And so these are probably light microscope stains, and when we stain them, we actually use a special stain that makes the flagella appear thicker than they actually are. It actually binds specifically to the flagella. This one I'm pretty sure it is an electron micrograph picture of these two, where they didn't actually stain it. They didn't do a flagellar stain per se for those. What did I want to say? Like I said, we're not going to stain them. So you're not going to see this. What else would happen to them when you stain them? You'd kill them and they wouldn't move, right? So we're actually going to use a technique where we're going to actually be able to see them move. So guess what we're not going to do? We're not going to stain them, right? So those techniques have some complications with them. Remember we've talked about before false positives and false negatives, right? Where it may appear to look negative, but in fact it's a positive organism, right? And in this case we're talking about motility. So if you overcrowd your slide, if you put too much bacteria on your slide, right? Whether we're doing the hang drop method or the wet mount, right? If you put too many bacteria, they're going to be crowded together. Under these conditions, would they be able to move? Not very well, not well enough that you would be able to see. So for a motile organism, you may look at it and see them not moving, right? And not necessarily recognize that they're crowded in, and that's why you can't see them moving. And so this would be what type of false result? This would be a false negative, right? Because you wouldn't see them moving, right? So you would term it negative, but in fact this organism is positive. So that would give you a false positive. Now if it's a non-motile organism, right, and you crowd them in, would that make them appear to be moving? If you crowd them in? No, that's not going to make them appear to be moving. And so in this case, would it have any effect on your results? No, right? You wouldn't end up with a false positive or a false negative, right? No effect. So this complication, you really have to worry about for motile organisms. You don't want to crowd them in because then you're not going to be able to see them move. And you may record the result inaccurately as a false negative. The other thing that can happen is they could attach to the glass slide. So for a motile organism, if they stick to the actual slide, are they going to be able to move? No. So you're going to end up with a false negative result again. Would this make non-motiles move? No. So would this complication have any effect on non-motiles? No. No effect. So we only again have to worry about attaching to the slide is if you have a motile organism, right, and it gets stuck and you don't see it moving. I have to say, this does sometimes happen, but even with the motile ones, sometimes they like kind of swirl around when this happens to them. The next one is a receding water line. And so what this is the water is moving. So just think about a log, right, in a river. The water is moving that log, right? It's floating down the river. Well, think about these organisms suspended in that water, right? They're going to move with the water, right? As the water is moving, they're going to move. So can the organisms move under these conditions? This is a tricky one. Yes. But the water is going to propel them more so, right? But again, they're motile organisms. So if you see them moving and you record it as moving, it really has no effect. The problem is on the flip side, right? Will this make non-motiles appear to be moving? Absolutely, right? And so you could end up with a false positive result. So you have to be, how do we avoid this complication? We have to be able to recognize when you have a receding water line. And so how you would recognize this, it should be in your homework for today. How you would recognize this is that everything will be moving fast in one direction, right? All in one direction, because it's going in the direction of the water, not the individual organisms. The individual organisms we'll see in a moment move independently of each other. Okay, so that complication, right, we really got to worry about false positives, non-motiles appearing to be moving. The next one, using an old culture. So are most of the organisms going to be alive or dead if you're using an old culture? They're going to be dead, right? So today, right, when I come, I'm going to want that new sample from last week, right? Our April 1st one, to be able to make a slide from that. It should be our freshest culture. So hopefully we have a lot of living organisms. Can the organisms move under these conditions? If they're dead, are they going to move? No. So you could end up with a false negative, right? If it's a motile organism, it's not moving, right? Or you don't see enough movement that you term it as negative. Now, will this condition make your non-motiles move? If they're dead. They're going to move when they're dead? No. So is this going to have any effect for our non-motiles? No. So what do we got to worry about for our non-motiles? The one thing, receding waterline, right? That's the one thing that's going to throw you off and maybe make them look like they're moving. For the motiles, we've got a couple of concerns, right? We want to make sure we're using a good culture where most of them are alive, so they'll be moving, right? We want to try to hopefully set it up so that they don't attach to the slide, and we definitely don't want to overcrowd them. Right? We don't want to take too much organism. So hopefully my videos will work. Oh, yay. All right, so I'm going to play it once. I almost wish I could turn off the lights in here, but it'd be too dark. I don't have the choice of just turning some off. So don't necessarily focus where the pointer is. It just, the microscope has a pointer. I want you to scan around for movement, where you think you see them moving. And then I'll play it again and we'll point them out. Yeah, we're gonna point them out. Okay. So you see right there, There's another one over there. It's kind of obscured. There's a couple that come in division right here. Yeah, up at the top. Good. Over here. See them right there? It's kind of twirling around. These are rod shaped, so they kind of flip sometimes. So for a second they might look like a cock's eye, other times they kind of look rod shaped. And all we did was decrease the lighting, right, to be able to see them, because otherwise they're relatively transparent. So we create a little bit of contrast by decreasing the lighting. So this one is almost over inoculated. You shouldn't have any problem seeing the organisms moving. No problem, right? They're all over the place. And again, if you focus in on like one, you can see them almost like tumbling. So to give you an idea of, what we're going to be looking for when we look at them under the microscope. I had some students that wouldn't stop talking while I was trying to record this. Extraneous background. All right, I think that's long enough. So there's a phenomenon that sometimes you'll see happening as well, and this is referred to as Brownian motion. And so molecules themselves will collide against each other and it'll almost look like vibration. You saw maybe some of that in that last one where there was so much stuff. So this is due to the thermal energy, right, that exists. So as you saw in the video, you want to really kind of focus in on an organism, and they're going to be flipping around, swimming around, right? They're not going to just look like they're vibrating, okay? You want to look for discern of movement. So why don't we stain them? Because we would kill them, right? And then they wouldn't move for sure. Although it helps with contrast, right? They would stop moving. So definitely no staining. We're also going to decrease the light, right? And this is because they're mostly transparent, and this is going to help us be able to view them. Yeah, the movement. So you'll notice how it was kind of shadowy and dark. And we have some, I don't remember if we
Part of that was from the book.
yeah, part of this is from our book.
Spherical aberration, and then we had to look up what that meant.
Yeah, so when we reduce the illumination by closing the iris diaphragm is one of the things that we'll do. This cuts down on something called spherical aberration of our microscope. And it refers to slightly inaccuracies of the focal point. So hence why it's kind of blurry, right? That's not my camera. It's because of the conditions that we put it under. Although it usually would improve clarity with these preparations because it's a liquid, right? And they're not stuck on the slide. This helps create the contrast that we don't get, that we won't get because we're not going to be staining. So it's difference between bright and not so bright. And probably the best explanation of this too is like, whenever you give a little kid a flashlight, what's the first thing they do? They shine it right in your eyes, right? That blast of light, you can't see anything. So again, if we let all the light in on our microscopes, there's nothing to absorb that light. There's no stain. And so you would just be blasted with all this light. Where if you're in a room, right, and you have like a night light or a little bit of light, you have all those shadows and such, right? I prefer pitch dark because then you don't get those shadows, right? When we're trying to sleep. So here we're trying to create shadows, right? So a little bit of light, we're going to create those shadows, we're going to create contrast that way. So why would you expect Brownian motion to increase the longer we observe a hang drop or wet mount? Well, Brownian motion deals with what type of energy? Thermal energy, right? What is that from? Heat, right? What on our microscopes can generate heat?
Light.
The light. And definitely on the microscopes that we use, I prefer to use the older ones. They have halogen light bulbs. They really heat up, right? And this room is hotter than the hinges of hell. So over time, right, you're going to have more heat energy, which would, and in this case, even dry out our slides. So we're gonna look at them as soon as we make them. So biochemical tests, right? What do they tell us about the functions, what our organisms can do? So there are three main pathways, right? We mentioned this before, for respiration. Glycolysis, where you can go from glucose to pyruvate. So we're breaking down that glucose sugar. And this can also, pyruvate and pyruvic acid are the same thing, right? They're used interchangeably by most instructors. The gateway step, where we're changing the pyruvate into acetyl-CoA so they could enter into the Krebs cycle, their citric acid or TCA cycle, has lots of different names. I usually prefer the Krebs. Give credit to the discoverers, right? So then we go into that cycle. We further break down the carbon that's present in glucose from pyruvate to acetyl-CoA, and all the carbon ends up being given off as carbon dioxide. That's how we eliminate the carbon. But why do we do all these processes? Why are we breaking down glucose? To generate energy, right? And the main way that it's done from these processes is we have electron carriers that carry electrons from glycolysis, from the Krebs cycle, from the gateway step, and they carry it to the electron transport chain to generate energy in a chemical form of ATP, right? When they add those phosphates, that's a really high energy bond, so it stores that energy until you break that bond later and release that energy, right? For whatever reason you need it. This is referred to as oxidative, and remember, oxidative and reduction, we're actually referring to electrons, right? So oxidation is the loss of electrons, reduction is the gain. So it's actually the movement of electrons through this electron transport chain in a membrane. For us in our cells, it's in our mitochondria. For most prokaryotes, it's actually in their membrane, right? Or any internal membranes they have. They don't have the organelle mitochondria like our cells do. So ultimately, carbohydrates like glucose, that's the most common one, are oxidized. We're removing electrons, right? We're generating hydrogen peroxide. In this process, some of the free energy is trapped in ATP, and the cell will use it later, right, for building molecules. Yeah. So aerobes, oxygen is what is accepting those electrons at the end of electron transport chain, right? It's a final electron acceptor, and we form metabolic water. Not all organisms can do these pathways. So here's glycolysis written out. And these are those electron carriers I was talking about. You should be familiar from biology class. This is a specific glycolysis pathway. This is the Ender-Duderhoff. No, this is the Enden-Meyerhoff. The Enden-Duer. Which one is it?
The blue.
The blue, right?
Is an alternative path.
Yeah, this is an alternative.
The structures are the regular glycolysis.
This is the usual, which is.
Which has a name and I forgot.
Yeah, it's. I said it a second ago.
Yeah.
You don't have to know it for this class. Aren't you guys glad? Because I can't even remember right now. I just said it. Enden-Meyerhoff. Yeah, Enden-Meyerhoff is the most common pathway for breaking down these molecules. So again, notice the electron carriers that are generated that are going to carry electrons to the electron transport chain. And then we enter into the Krebs cycle, right? We further break down these molecules. Citrate is recycled. That's another reason why this is called the citric acid cycle sometimes, because the acetyl-CoA binds with the oxalatate and forms citrate. And then that's further broken down through the process of several reactions. There's not showing all of them here, right? We do get some ATP produced, or GTP, at the substrate level, right at the enzymatic level, but all the rest, right, are generation of these electron carriers, right? They grab the electrons from the breakdown of these molecules, and they're going to head off to the electron transport chain. So there we have carbon dioxide. So tricarboxylic acid, another reason for that name has to do with how many carbons, right? So you have 3 groups of carbons right here in citrate. 2 groups of 3 carbons. 2 groups of 3. You see? 2 here, 2 here, and 2 here. So that's another how it has that name. So here's our electron transport chain. So these electron carriers are going to pass them through a set of carriers in the membrane, right? And they're going to pump hydrogen ions from one side of the membrane to the other. That's why it needs to be in a membrane. And here's your other electron carrier. So pump in the hydrogen ions. You'll see NADH pumps more than FADH. So therefore you actually generate more energy from NADH than FADH. These molecules in the membrane create an opening for it to move from one side to the other of the membrane.
Pores.
Pores, yeah. And then, so once they're on, oh, that was an important point there. I skipped over it. So the protons are pumped inside the prokaryotic cell, or in the case of the mitochondria matrix, to the outside of the membrane, right? So then, so here's that matrix, and you'll see them being pumped across that membrane. But then when they travel back through this another pore, but this is also an enzyme, ATP synthase, so it adds the phosphates to the ADP to form the ATP. So it creates a gradient, almost like water, right, can be used to generate energy to turn a turbine. In this case, we're using the hydrogen ions, or pumping across. And so, again, it's oxidative phosphorylation. So we're adding the phosphate by using electrons, or movement of electrons, to generate that energy. In this process, the one main reason why we're talking about this is that sometimes the electrons combine with oxygen and they can produce superoxide. This is a highly reactive, very damaging molecule. So remember here, if it goes like it's supposed to all the way down the pathway, it should form water, right? The oxygen combines and we form metabolic water. But organisms that utilize this pathway, there's the chance that sometimes they're going to produce this highly reactive byproduct, right? Superoxide dismutase. So we have, superoxide, excuse me. The enzyme we have to eliminate it that we'll talk about helps remove it. And another one that can be produced is hydrogen peroxide, right? Which is also as destructive, but not as destructive as superoxide, right? So these are two unfortunate byproducts of aerobic respiration, right? Where we're using oxygen to accept these electrons at the end of the electron transport chain when we break down glucose. So the catalase test, right? We are going to determine if we have the ability to break down one of these byproducts, and that's hydrogen peroxide. So hydrogen peroxide was our reagent that we used. If the organism has the enzyme, which is catalase, you can break it down into water and safe gaseous oxygen. And so how do we know that gaseous oxygen was produced? What did you guys see? Bubbles, right? So, and it's also why hydrogen peroxide is kept in that dark bottle, because light can cause this reaction to happen too, right? So why it's in the dark bottle. Over time, right, that reaction can happen and eventually you just end up with water. This is why definitely we want to test this reagent, okay? And don't believe in the old wise tale that if you put hydrogen peroxide on your wound and it doesn't bubble means it's not infected. And you are catalase positive. You are an aerobe, are you not? Your cells, when they're damaged, release catalase, this enzyme. You should always, always, always bubble, right? If it doesn't, it means you just poured water on yourself. Your hydrogen peroxide's old. Go get some new stuff. But even better, right? Because this can damage even your own cells, they suggest not using hydrogen peroxide as an antiseptic anymore. Soap and water and a little triple antibiotic ointment for minor wounds, right? Because this can be damaging. You can use this to clean a whole bunch of other things, right? Clothes, household items, earrings, right? All kinds of things, right? Don't use it on your skin anymore. But if you happen to pour it on your skin, it should always bubble. Make sense? All right. So breaking down glucose in the presence of oxygen, right? So we're going to use these electron carriers and generate some ADP, ATP, and GTP. We're going to generate water, right? We're going to also, as a byproduct, have carbon dioxide, and that's where the carbons go. So carbon is oxidized, we remove the electrons. The coenzymes gain the electrons, they're reduced, right? And we also have some substrate level phosphorylation. And then in our electron transport chain, if you were to add it all up, it should yield about 38 ATP in bacteria. In eukaryotes, we lose a couple because of that movement out of the mitochondria, out of that organelle. Of course, this is theoretically, right, how much we should get. It's not an actual yield. And then we have 4 that are produced from the substrate level. The rest are what are produced in the electron transport chain, the oxidative 34. So again, we can get a total of about 38. That's a lot of energy, right? A lot of energy. And as I said, typical yield is a little bit less than that. All this information is also summarized for you in your appendix on page 529. So respiring organisms manage to harvest approximately 30% of the free energy from glucose, right? So they could generate about 2,880 kilojoules per molecule. Each ATP generates about 30 kilojoules per molecule.
Actually, can you go back one slide? I just just wanted to point out that, you get 4 ATP substrate level phosphorylation. So if you don't have oxygen, you only get 4 ATP out of glucose. If you do have oxygen, you get an extra 34.
Right.
So that's why in the FTM experiment we did, they're so much better in the presence of oxygen.
Right. So the reason for all the numbers, right, and the differences between substrate level and oxidative is that you get a lot less energy, right, if you are stuck at substrate level. If you can't do, we don't have that slide coming up? I thought we did. Okay.
No, I know.
Oh, okay. I thought we did.
That's the point I wanted to make for that slide.
Yeah. So I want to go back to this other one.
Actually, you're going to a slide I wanted to revisit this on.
Yes. Okay. So for this one, right, for glycolysis, right, we're generating some energy here. When you go to the Krebs cycle, you're producing so many electron carriers that if they can't go to the electron transport chain because there's no oxygen to give those electrons to at the end, then this process stalls, right? You can't have these carriers having no place to bring the electrons, right? You only have so many of them. They're all loaded down with electrons. They're just sitting there because they can't give them to the electron transport chain and then come back to the Krebs cycle and glycolysis. So you get stuck in just glycolysis, right? And you can't move further down because there's no oxygen. And so we had one type of organism, do you guys remember, that I want to go all the way to the other one, to this one. So you would be stuck with around 4 ATP from just substrate level, where 34 come from the oxidative, right, from the electron transport chain. So you guys remember which organism could grow in the presence of oxygen and grew faster because look at how much more energy it was producing, as opposed to without oxygen, it grew slower because it had less. Facultative anaerobes, right? Facultative anaerobes. And so this is why you see that difference in growth, right? It's what processes can they do, how much energy does it actually generate. So here's some great videos. If I don't already have the links to them, if you want to actually see this stuff moving, little animations, these are cool ones to refer to. If an organism lacks the electron transport chain, how does it recycle and reduce the coenzymes NADH to NAD? So where does it drop off its electrons? If it doesn't have it doesn't even have an electron transport chain. Never even mind not having oxygen to accept the electrons. What if it just doesn't even have an electron transport chain? They got to drop them off somewhere, right? Go back where it came from, then we're just regenerating glucose, right? The answer is we're going to oxidize, right? Reduce. We're going to give those electrons to organic molecules, and we're going to form acids, alcohols, and gases. All of that is termed fermentation.
And that's when the growth is very slow.
Yeah, and that's why the growth is slow, right, under those anaerobic conditions for a facultative anaerobe, because he's doing fermentation, right? He's just doing glycolysis, that's it. And then he's given those electrons back to products that he's broken down from glycolysis. So here's pyruvic acid, right? This is our byproduct from glycolysis. We can release carbon dioxide and form formaldehyde. Some ones will continue and add the electrons back to the formaldehyde and produce ethanol. It's an alcohol, right? Brewers or baker's yeast, Saccharomyces, a yeast, actually does this process, right? We love it for being able to do that process. This is the ethanol, right, the alcohol that we drink. Another option is, again, releasing carbon dioxide and forming formaldehyde, and then adding another pyruvate to it and forming alpha acetolactate. I was so gonna mess that one up. Acetolactate. Adding hydrogen ion and releasing some carbon dioxide, and we can get the formation of acetone, which some of our organisms do, that you guys have. And then again, we're dropping off that electron carrier from earlier, and we're forming this alcohol, 2,3-butanol. And this is a rate limiting step, which means that sometimes you're going to have not so much of this alcohol. That's the best way to explain it.
Because it's rate limiting, you have an accumulation of that intermediate acid.
Yeah, we get an accumulation of this intermediate acetone. Like, it isn't really quick going from here to here, that process. So our MRVP tubes, we ran 2 different tests, right? The VP, the Vogts-Proskauer test, detects the intermediate acetone right here, which accumulates in our media, right? So that's actually what we were looking for the presence of. And I think you guys have some red tubes finally. Huh. We got some positive tubes that we didn't have positive before. For the VP. Is yours finally positive, Mohammed? No?
Only mine.
Did mine turn out positive over there for my positive control?
Mine's turned
VP.
red.
See where your VP is. Positive and negative look different. Hold them up, those two. Oh yeah.
Oh yeah, you can definitely see the red. Yours is completely red.
Yeah.
Maybe I have yours confused.
Acetone. What was it? The fermenting.
Okay.
Glucose.
So more fermentation. So pyruvic acid can be, add the electron carriers, add the electrons back to it, and produce lactic acid. Lactobacilli, also another bacteria in this case that's used in the bread making process for sourdough, right?
Mm-hmm.
Yeah, for sourdough bread, because it's acidic. And also for us, under anaerobic conditions, right, where you don't get enough oxygen flowing when you're exercising, we say the buildup of lactic acid, right? And this can be toxic. You can further add electron carriers to it and release water and produce another acid, propionic acid. No organisms have evolved to do this, though. They do it a different way. They actually add carbon dioxide to pyruvate and get oxalate, which then is reduced to succinate. This is another acid. And it's actually 3 steps in the Krebs cycle in the opposite direction, right? Enzymes that are causing the formation of this acid, and then they convert it into propionate.
So it seems like a lot of work for
Yeah.
an organism to do that, but I think it evolved because they just reutilize Krebs cycle.
Right,
That's
so
right.
even though, it'd be shorter to go from here to here, they have the enzymes already from the Krebs cycle, just going in the opposite direction to produce these acids. So, a couple more. So, pyruvic acid to acetyl-CoA, right? We're picking up electrons, and then it would enter into the Krebs cycle, right, or TCA, and it can produce lots of different, and there's a whole chart of it, acids, alcohols, and ketones. Pyruvic, so these are all products of glycolysis, right? Pyruvic acid, and then the fermentation process of getting rid of those extra electrons. So formic acid can be produced. No oxidation reduction here, though. And then the acetyl-CoA can enter into the Krebs cycle if it can. Our phenol red test detects the presence of gas. That's why you have the little tube in it. And acid, because phenol red is actually a pH indicator. And because it's a pH indicator, it not only can detect acid, but another thing that can happen, which is the deamination of proteins. The methyl red test that we ran detects mixed acids, and as you saw with all these fermentations we talked about, a lot of them were acids, right? And so some of them produce multiple acids, so much so that the pH gets very low, right? And that's what our methyl red test tested for. So lots of acids, right? So formate, which is a 1-carbon acid, acetate, 2-carbon, propionate, and lactate are 3-carbon acids, butyrate and succinate, 4-carbon acids. These are all possibilities for your organisms depending on what pathways it's using. Alcohols, methanol, ethanol, isopropanol, butanol, and then the one that we're going to look for today is the ability for them to make 2,3-butanol. But again, we're going to be detecting the intermediate in that, which is acetone, and which is different from acetonin, which is a ketone that can sometimes be produced from fermentation. And then gases, your possibilities, as you saw, lots of carbon dioxide, right? It doesn't distinguish fermentation from respiration, though, right? So if an organism is respiring, they'll still produce carbon dioxide, right? But hydrogen gas is unique to fermentation, is only produced under fermentation conditions. And today, you're not going to be able to tell which gas or if both were produced by your organism, because all you're going to see is a bubble, right, indicating that gas was produced. We don't know which gas it was. Do I need to read through this, Peter?
No.
Okay.
It's a point of trivia that's interesting.
Okay. Yeah. Okay, so mixed acid fermentation. So which test out of our 2 tests, methyl red and Vogus Proskauer VP, which one actually tests for mixed acid? So which one is a pH test? Methyl red. What's the reagent? This one's easy. Methyl red. It's the same name as the test. What do the products of this type of fermentation do to the pH of the media? Right? So we're talking about acids, right? So they're going to lower the pH. Yep. Everyone still with me? Okay.
There's a couple of extra slides that may be throwing you off here, Erica.
Okay. Oh, yeah. Oh, okay. Yeah, that's okay. Nope. So what is the measure of pH? It's actually the measure of hydrogen ions or protons, right? The positive component of an atom. A hydrogen atom is just a proton and an electron. That's it. So when you have these protons being released, right, it's hydrogen ion, it's referred to as. These are very significant because they interfere or they make chemical reactions possible, right? These movements of electrons and protons around. So we pay special attention to hydrogen ion presence, right, in a solution, right? Because a lot of times when you add something to solution, they're going to release these hydrogen ions. These could either interfere with chemical reactions or aid them. It depends on what is necessary for that chemical reaction to move forward. And it's actually the negative log of the concentration, right? M referring to the molarity. So 4, right, a pH of 4, which would be acidic. Remember, 7 is neutral, right? We have an even balance of protons to ammonium ions, the other half of the water molecule, you could say, the OH. So notice that the concentration would be 10 to the negative 7, right? But the pH is the negative log, so it's just basically the exponent, right, of this concentration. And so that's neutral. Anything below 7 all the way to 0, right, is acidic. Anything from 7 up, so 8 to 14, which is the scale, would be basic or sometimes referred to as alkaline, right? And so in this case, lots of hydrogen ions, very little hydrogen ions, okay? So different pH indicators have different ranges, and they change color in those ranges. So even crystal violet, look, a dye that we commonly use, it'll have differences in color depending on the range in the pH scale. So ones that we're using, here's methyl red. Oh, click, we got them circled. So there's our phenol red. So one of the reasons why it's kind of called that is that it's mostly red near neutral pH. You see that? And then it's gonna, when it goes below neutral, it's gonna turn yellow. Bromothol blue, right? The pH range that we use it around neutral, it's green. This is the one that's in your citrate tubes, those slanted tubes. So that's why some of you guys have green tubes, and then some of you guys notice the pH went up, right? And that's why you have blue. Here's our methyl red. So notice very acidic to get that red color, right? 4 and 5 range, right? You get that color change. And then even still acidic, right? But not very acidic is going to be that yellow. So that's just the ones that we use. Yeah. So 2,3-butanol fermenters, right? So this is an alcohol. Which test? Well, by default, we already talked about methyl red, so therefore this test, VP, Voss Pascal, is going to tell us if your organism can do 2,3-butanol fermentation. If when it's given, right, sugar, glucose, does it convert it into 2,3-butanol through fermentation? So we used 2 reagents, right? Barrett's reagent A, which is actually alpha-naphthol. So I guess it's an alcohol, huh? Oh well. No?
Naphthol is a organic molecule.
Oh, it's an organic.
It kind of looks like a purine to me.
Yeah.
It's complicated.
Yeah. Where reagent B, which is why we were very cautious, is definitely a strong base, which is potassium hydroxide. But remember, even though we're wanting to know if your organism can do 2,3-butanol fermentation, we're actually not testing for the presence of the 2,3-butanol. We're testing for an intermediate. And what is that intermediate? Acetonin. Which is different from acetone. It's acetonin. Okay, so glycolysis, as we've been looking at, right? You can break it down into pyruvate, add in acetaldehyde and form acetate, acetolactate, excuse me. And then we have our acetonin, which can be broken into diacetyl or 2,3-butanol. We're actually testing for the acetonin, so notice that the acetonin binds with the alpha-naphthol. And the second reagent is the potassium hydroxide. And what else do we need? Oxygen. Right, and this is why we had to make sure to shake our tubes really good after adding the second reagent. And then diacetyl is in the media. Diacetyl forms and then binds with guanine, a peptide that's in the media, and it produces the red color. So for our methyl red test, right, it dictates a positive result. Red, right? And again, that denotes a really low pH, right? Brown, it's very acidic. And then we have our VP test. That one is also red, although my pictures are kind of pink. Oops, okay. So as you can see, nice positive results here. This one's a little bit of a weak positive. Do you see how it's light? And this one, see how it's discolored, but it is not a reddish color, right? So it is not positive. For each of these tests, each group, right, and we already said this, so we know our reagents are working, right, and we get the expected results, and we can compare our unknowns too. Did that already. So in this picture, as I said, notice I didn't have a true red. Do you guys have a really red color hopefully this time? Okay. Yeah. Okay. What would a false negative for the VP test more likely be attributed to? Poor sensitivity or specificity of this test. Remember we talked about this before, the difference between specificity and sensitivity? So is this test very specific? Yeah, it's a chemical reaction that's going to bind with acetone. So it's not specificity, but sensitivity, right, is our problem here. And what factors into the sensitivity? What do we have to be able to see to know if this test is positive or negative? We got to see a color change, right? And so there has to be enough color that's detectable by our eyes, right? And so we have to have enough acetone to react with the reagents to get enough of a color change. So the reason why we can run this test is because acetone isn't quickly turned into the 2,3-butanol. Remember we said there was a rate limiting step there, and so we have enough acetone that we actually are able to detect. So why were you told to shake the VP tubes after reagents were added? Right, we're adding oxygen, we're doing that, and we need that for this reaction to proceed. So that's one way you can end up with a false negative result, right? If you don't get enough oxygen in there to make it a positive result. Why is the methyl red test red immediately? What are we actually testing for? What is methyl red? A pH indicator. We can read those immediately, right? You don't need a chemical reaction to happen. For the VP test, though, what are we waiting for to happen? A chemical reaction, right? And sometimes those take time, right? So we're actually waiting for that chemical reaction. So as we saw, as we looked through some of the fermentations, this is for E. coli. And notice it's under specific pH indication, I think, for this one. Notice all the acids it could potentially produce. Lactate, acetate, formate, succinate, also hydrogen gas, and carbon dioxide gas, right? So this is a very strong acid producer under fermentation conditions. And so you'll definitely see that, right, for organisms like E. coli, right? Lots of acid production detected by our methyl red test. Some microbiologists recommend re-incubating the organisms that produce a methyl red negative results for an additional 2 to 3 days. Why would you think that would be helpful if we ended up being negative? We just incubate it longer. What happens as they're growing? They're fermenting, right? And what are they producing when they ferment? Acids, right? And we need to get to a level where there's enough acid that it's detectable by our pH indicator. So the longer they go, are they going to produce more acid? Yeah. So our methyl red test, actually 2 drops. We did 3. It's okay. It's just a pH indicator. Right. So orange or yellow is not red, therefore it's negative. But again, right, if it was orangey, indicate that, right? I'm not going to read through the scientific method for this one, although I should. What? Okay. You guys will have your power. Yeah. We got to get going. All right, so these are your phenol red possibilities. And so I have the blue rack on your guys' lab bench. Can you guys find it? Find those tubes. We're going to reference those so you actually have them in front of you. And so you'll notice that they're labeled, right? AG stands for acid. And what color is that tube? What color is it? Yellow. And the gas, can you see the bubble in the little derm tube? This one has a lot of bubble. Those guys might have just a little bubble at this point. Should have done fresher tubes. Alright, and then the code that we use for acid, no gas production. So acid, again, what color is it going to be? Yellow. But you shouldn't see a bubble in the tube. We code that A minus, right? A for acid, negative sign for no acid production. You should have a negative control in your rack, which will be maybe kind of orangey. It says uninoculated, which means we didn't put an organism in it. So you'll notice in some of those tubes too, when you're looking at them, you may see some turbidity, some growth of the organisms, right? So we know something grew in it. If you have turbidity, but you don't have any color change from your negative control, it means your organism grew, right? But it didn't produce any acid. Or the other thing that can happen is that alkaline products are produced, and we term that K, and it should be a nice bright pinkish magenta tube in your rack labeled K. And this is when they eat the protein and produce alkaline products. And we label that K. Holy cow. Oh, yeah. So the base broth, right, has phenol red in it. It helps us, it's a pH indicator. There's lots of different sugars that we could add to these tubes, right? And they come pre-added. And the ones that we have, it's actually glucose.
With the button.
Right? Lots of different available. We're using glucose. So what are the common byproducts we've already said of pyruvate fermentation? So general. No. Fermentation. Fermentation of pyruvate. Acids, alcohols, and gases, right? And the gases could be carbon dioxide or hydrogen gas, right? We looked at one alcohol so far, 2,3-butanol, but there are others like ethanol, right? And several acids. Which one of the three cannot be detected with this test? So the phenol red test, which one are we not detecting? Or let me put it this way, which ones can we tell your organism produces? Can we tell if your organism produces gas? Yes, because of the bubble in the derm tube. That's the name of the tube, the little tube that's upside down. Can we detect alcohols in this test? No. Can we detect acid? Yes. That's what our pH indicator helps us do. So again, there's a pH indicator. The pH indicator is the name of the test, right? It's phenol red. And this is its scale. So around neutral, it's going to be orangey red, right? Alkaline, it's going to be that pretty magenta pink-like color. And acid, right, it's going to turn yellow. Right. So here's all the different variations that we could see. What color does the media turn if acid products are generated? What color? Yellow. Yellow. What color does the media turn if alkaline products are generated? Pink or magenta, right? So we can tell the pH of the media just by looking at the color because of this pH indicator called phenol red. What does the derm tube help us detect? Gas. And again, remember, we don't know what gas. We don't know if it's hydrogen or carbon dioxide or both. If the glucose is fermented, what happens to the pH of the media? Well, what's a product of fermentation? Acid, right? So it would be acidic. Lowers the pH. And we're going to end up with yellow, right? Peptone is a mixture of proteins and polypeptides in the media component that some organisms can use as a carbon and energy source. If the peptones are their amino acids are deaminated, this produces ammonia. And what will happen to the pH if you have ammonia produced? It's going to go up, right? It's alkaline or basic. And so again, we're going to get that magenta color. So we'll actually be able to tell what your organism is eating, what processes it's doing. Is it fermenting and producing acid, or is it deaminating the protein sources and producing alkaline products just by the pH color? So when scientists sometimes do this, they would do a broth tube that doesn't contain any sugar. And then they would do your test tubes that actually contain the sugar. So our tubes have a choice, right? Because we have the sugar and the protein in the tube. So what happens if they eat the glucose? It's going to turn acidic. What happens if they eat the protein? The pH is going to go up, right? And it's going to turn pink. What happens if the bacteria eat all the sugar and then begin to eat the protein? So if they eat the sugar, it's going to turn what color? It's going to turn yellow. And then as they start eating the protein, the pH is going to go back up, right? And so yellow, right? And then it's going to go up higher. So this is hard to predict. It depends on where it's at. And if you look at, and then protein only, they could only eat pink. We should add the pH scale in here. Let me grab it. There it is. Okay. Oh, I got to end to do that. Copy. So I have some examples of that I'll pull out. Okay. So, let's see.
I don't know where you got that image. I'd like to get an image that actually has the little pH, what's it called? The little strip, pH strip that the alkaline in.
Yeah.
You know, it's always showing it.
Yeah,
acidic.
acidic.
We need a picture like this with the alkaline.
Yeah. So it's going the other way now, right? So it's yellow and it's going to go up. So it'll go through orange, right? And then it could go all the way to pink depending on which way it's how far down it went and how far up it will go. I have different phases of that I'll show you guys. And one of your guys' organisms does do this. It within the 48-hour recommended incubation period. So I had to take it out and put it in the fridge to keep it from going completely pink. And yours I forgot about. So yours went pink, right?
Hold it up.
But one indication, find your organism. Yeah, see how it's pink? But do you see, like in the tube, it's kind of, do you have any gas or anything? No? No. But hers, I forgot to take out and let it go too long. It's one of those ones that will start, when it runs out of the sugar, start eating the protein. Okay? Huh? Yeah, it might be in the refrigerator. Might be in the refrigerator. So I'll grab them out for you guys. In the interest of time, I'm going to go over this table next time. Because I don't think we quizzed them on this, right, Peter? I'm just skipping this table, y'all. I'm not skipping everything. We'll come back to this one. I just want to get to the, well, we do have to go over these. Okay. So if you have turbidity, right? It's a slow grower, right? So it still looks like the uninoculated control, right? This would be termed a false negative because the organism hasn't grown enough to produce enough acid, right? So what would we attribute this to? Would this be poor specificity of the test or poor sensitivity? If we don't have enough of acid to produce enough of a color change, it would be sensitivity, right? It takes a lot of acid to produce that pH. Early formulations of the media had smaller carbohydrate sugar amounts, right? And usually would produce false alkaline pink results after 48 hours. Why do you think this happened? So what did they run out of? Sugar, right? And then they started eating the protein. But then one of the main reasons why we do this test is to know if they ferment the sugar, right? We want to see that. So what's one way we could fix this? What 2 factors could we change? So they're eating up all the sugar really fast because there's a small amount. So what could we do? Give them more. Give them more sugar, right? Or we're also incubating for 48 hours. What could we do to the incubation period? We could shorten it, catch them in the act, right? Eating the sugar before they get to the protein, right? So we could add more sugar or incubate for a shorter time. And of course, we have more sugar added. The problem is they're eating the protein because they ran out of sugar. The question we're asking with this test is, do they eat the sugar, right? Not necessarily do they eat. We also are finding out whether they eat the protein or not, but we're most concerned with, do they ferment the sugar first? Okay, so the citrate test, and this is your slanted tube, so on the other side of your rack you see 2 tubes. This is testing for the presence of, and I have an example of the sucrose transporter here, but this one is for citrate. And what permease proteins are, they're proteins embedded in the membrane that specifically transport certain molecules back and forth, right? So in this media, there's one carbon source, citrate. That's it. If they don't have this molecule, this citrate permease in their membrane, they can't bring it in. So guess what's going to happen to them? So the only carbon source is citrate, sodium citrate. The only nitrogen is ammonia dihydrogen phosphate. Since organisms need both a carbon and a nitrogen source, they only have one choice here. What will happen to the organisms if they can't use these molecules? If they don't have citrate permease to bring in citrate, they die. They die. So there's a pH indicator in this media, right? It's bromothiamyl blue. Bromothiamyl blue. Thymol. Bromothiamyl blue. This one, as you can see, as our uninoculated control, we start out around almost neutral pH. So your media was green to start out with, right? So the uninoculated control in your guys' blue racks, notice, is green. Then, as your organism, right, utilizes the nitrogen source, it'll change the pH of the media. And in this case, right, the ammonia that's utilized is going to yield what color? Blue, right? Because it's going to pull away the hydrogen ions from the media, right? And it's going to turn blue. If the media is green and you see growth of the organism in the tube, is this a positive result for the utilization of citrate? What do you think? Yes, because the organism can only grow if it's utilizing that carbon source. But I'll tell you guys that for your tubes, if you see what may be growth, you may have put too much bacteria on there. And these tubes have gone long enough that even if yours is a slow grower, your tube should be blue if it's positive. If it's not blue, it's probably not positive. It's probably negative. So again, citrate positive organism may not produce color. Is this a false negative result? Yes, right? Not enough ammonia to produce. So if we're dealing with ammonia, right, we're dealing with a color change. Is this poor sensitivity or poor specificity of our test? Poor sensitivity, right? Okay, catalase is our last one, which we already have our results for, but let's talk about this. So flavoprotein is an electron carrier that passes electrons to oxygen. But remember, this is one of those steps where we could get those bad byproducts produced. So we have enzymes to protect us. Aerobes have superoxide demutase, which is going to convert that superoxide radical into safe oxygen and hydrogen peroxide. Catalase takes that hydrogen peroxide and converts it into water and gaseous oxygen, which is why we see what? Bubbles. Right? So on our slide test, and remember, we had to put the bacteria on the slide first and then add the hydrogen peroxide. What was the reasoning for that? So would a false positive reaction between the inoculating loop and the hydrogen peroxide be cause poor specificity or sensitivity? Specificity. So the problem and why we do the procedure the way we did is the loop itself, the metal of the loop, can sometimes cause hydrogen peroxide to bubble. So that's why you put your organism on first and then added the hydrogen peroxide, so that we didn't have a specificity issue. We weren't questioning, was it the loop or was it our organism that was causing the bubbling? And then I think we have one more. So also for some of you guys, and it may have to, I know because we use a pretty fresh culture for most of you guys, you may have a weak positive when we did the slide test, right? And one of the ways to kind of help you see the bubbles is you could put it under the microscope. We didn't bother doing that today because we were going to do it right on the slant itself, and usually you get lots of bubbling because you have lots of organism, right? So this difficulty of seeing the bubbles and thinking yours was negative, right, which happened for some of you guys, you didn't see your bubbling on the slide, would it be poor specificity of this test or poor sensitivity? Poor sensitivity, right? Difficulty reading a positive result because the difficulty of being able to see with your own eyes those little tiny bubbles. Oh, no, we just still have one more? Oh, yeah. We already answered this, though, before, so you guys should know. Why is it advisable to perform this test on a known catalase positive organism along with your unknown? Make sure that the reagent is good, right? That it's not water, right? That we get the result we're supposed to get. And then why did I do it in the uninoculated tube? What's in that tube? Media, right? Uninoculated means I didn't put any bacteria in it, so it's just media. And we need to factor that out, that it does not cause the bubbles, right? So we don't get false positive results. It's another control to factor out the presence of the media when we do the slide test. I mean the other one. I think this is it, just the scientific method, yeah. All right.
There's one slide I wanted to go back to just briefly. It's beginning of the Krebs cycle. It's a green comment in the lower left corner.
Krebs cycle. I'm not gonna worry about it right
Okay.
now.
All right.
We're like beyond time.
Yeah, right.
Let's see.